Welcome everyone to this panel discussion as part of the BGE Day, the Biodiversity Genomics Europe Day at BG 23. So I'm Pete Hollingsworth from the Royal Botanic Garden, Edinburgh, and I'm one of the coordinators of the Biodiversity Genomics Europe project. So just starting with a recap for any of you who are joining us partway through the day. What's happened so far? We've had an introduction to the European landscape and priorities from the perspective of the European Commission around biodiversity and biodiversity genomics, as well as a summary of the aims and objectives of the BGE consortium. We then had a series of excellent case studies catching some of the many different applications of genomes and barcodes for understanding and characterizing biodiversity. And we've just now recently finished a great session, including invited speakers highlighting technological innovations and logistical opportunities for increased utilization of genomic data for biodiversity characterization and conservation. So if you've missed any of that, it's been a great day so far, and I commend those earlier sessions to you on the conference catch-up service. So now we're into this final session of the day, which is a panel discussion. And here the theme is exploring what do we need to do to accelerate the integration of biodiversity genomic data into the evidence base that's needed to meet stakeholder needs? What do we really need to do to maximize the impact and usefulness of biodiversity genomic data? And of course, this is at a time of a massive global biodiversity crisis. So we've got a great panel assembled here, and it includes a whole range of expertise. So researchers with expertise in applying genomic data to practical conservation and sustainability projects. We've got panelist experience in the commercial deployment of DNA biomonitoring, and also perspectives from both the national scale for deployment of DNA biomonitoring and that from an international intergovernmental agency as well. So as we go on during the session, please submit questions via the Q&A function, and where appropriate, direct those to the individual panelists if it's relevant for a particular panel member. Okay, so to start proceedings, I'm just going to kick off with a round of introductions from Magda, Elena, Pedro, Charlotte, Tiina, and Michael.
Thank you, Pete. Hello, everyone. I'm Magda Kharrat. I'm from European Forest Institute, based in Barcelona. I'm professor at St. Joseph University in Beirut, and I'm actually working as coordinator of one of the biggest ecosystem restoration in Europe, forest ecosystem restoration, where I will be commenting on the need of biodiversity genomics in our field of forest restoration.
So hi, from my side, I'm Elena Buzan. I'm coming from University of Primorska, Slovenia. And in BGE project, I am leading the task Genomic for Society. And in this task, we try to bridge the gap between the researcher working in applied genomics, conservation practitioner, and the public.
Good afternoon. I'm Pedro Beja from a research center in Portugal called CIBIO. I'm one of the team members of BGE, responsible for the application pillar, but I'm mostly an ecologist with a background in ecology without molecular, but now increasingly using the molecular component. And I'm also a member of the Europa-Born project, which is relevant in this particular context because it's a project commissioned by the European Commission to design a network of a European biodiversity observation network, which could benefit a lot from the molecular component.
Hi, hello everyone. My name is Charlotte Germain-Aubrey. I work at the Secretariat for the Convention on Biological Diversity under UNEP, and I am the Program Management Officer supporting parties to the CBD in their work towards a global solution for the sharing of benefits that come from using sequenced data globally. Thank you.
Hello, everyone. My name is Tiina Laamanen, and I'm coming from Finnish Environment Institute. And currently, our institute is running quite many eDNA-themed national level and international level projects. Last year, we published National eDNA Roadmap, where our aim was to develop a plan how we can proceed with these molecular-based methods and how we can implement these methods into our environmental monitoring programs.
So good afternoon, everyone from Austria, Innsbruck. I'm Michael Traugott, and I'm based at the University of Innsbruck as a professor of applied zoology, and I'm using DNA-based methods to study biodiversity since many years. And besides doing research, I'm also a co-founder and the current CEO of a company called Synsoma, which offers this type of DNA biodiversity monitoring services to a lot of clients within the environmental sectors.
Thanks all, and it's wonderful to have you here forming the panel. So just before we open for questions from the floor, and again just reiterating that encouragement to put questions in as we go along, what I'd like to do is start off by drawing on this very kind of wide range of perspectives that we've got from our panel, by inviting each of the panel members to comment on what you see are either some big opportunities or on the other hand, barriers to overcome, so that DNA and genomic data are more effectively used for biomonitoring of biodiversity and conservation. So over to you, Magda, first. Sorry, you're muted. There you go.
Yeah. From my first background as a researcher, I was working on producing some data about barcoding animal and plant species for restoration. So understanding a little bit the web, which animal is eating what, who is dispersing what from a genomic perspective. But then one of the main obstacles was the reference data. And this is something that the first block at some point. So as a researcher, we would like to have access to available data that are not shared through databases. And I think this needs also kind of a framework where we can ensure for every researchers where the data can go and where we can access this kind of data. And then to make, when these data are available, we should ensure that the policies or the policymakers knows about those tools, so they can include them as a request in their monitoring. I mean, when they request monitoring, they know about these tools and they ask for it in their. So this is my first response, and I will comment later on specifically on ecosystem restoration in Europe, how we can use this.
Perfect. Thanks, Magda. Elena?
Yeah, and maybe I will start with the experience we have in European Reference Genome Atlas with the researchers, and I will explain first research view. So we were quite surprised when we realized that people working in genomics does not recognize the stakeholders or the people who will use the genomic data. So first of all, we need to work in this aspect to see the people who are working in genomics, they do know how their work can be useful. Another aspect, it's also very important for them and for all of us that we are aware that there is a limitation of genomics and that we need to overcome this limitation by using other data like a field survey, like even knowledge, I would say traditional knowledge, citizen science, and many other aspects, because we cannot solve everything by genomics in conservation and bioeconomy. On other aspect, we have a stakeholders, and what we realize until now, many stakeholders, there's a really huge gap in the knowledge and also what are really genomic data can be useful. And also today we heard a lot of how the genomic data can overcome the genetic questions connected with the genetic load. We cannot solve this with the genetic data, and we still have some kind of stakeholders. We do not recognize even the differences between genomics and genetics and many other things, which are now we are facing with all of this huge knowledge in genomics.
Thanks, Elena. And now over to Pedro.
Yeah, where to start? There's so many things that we can say. I think one important thing is that we should realize that there is a long distance between having a nice method to publish papers and a nice method to do standardized monitoring over the years and to inform policy. And people from the research side usually thinks, and I think too, so that once we have the paper published, our work has ended, and this is it. Someone will pick it up and will apply it at scale for monitoring and solve all the problems of the world, and that's not even close to that. And I'm sharing a little bit my experience with Europa-One over the past 3 years, where we have tried to start designing an European biodiversity observation network in Europe. And we have gone through identifying what's the status, what are the gaps, what are the bottlenecks. And we found there's very little thing similar to what might be a biodiversity information network across Europe. There's very few methods that are applied consistently across the countries. Then that information is lost in the archives of national agencies, and when it flows to a central processing unit, then it's difficult to access the data. And so over these years, with my two hats of BGE and Europa-One, I've tried to bridge a little bit the gap. But I think, and it's obvious that there are huge gaps to be crossed. One is from the genomic side, we need to know what is needed for practical application. We tend to do our research irrespective of what the stakeholders actually need and what do they want for regular monitoring. And on the other hand, the stakeholders need to know what they should ask for, because they don't even know some of the approaches and some of the techniques that now exist. So for not talking too much, because there will be many things to say, I concur with Elena and Magda that bridging this gap between the research and the actual implementation, going closer to what stakeholders need, both to answer to their queries and to let them know what is now possible to do in a relatively cost-effective way. But then there are many other problems that we can talk later.
Perfect. Thanks, Pedro. And now Charlotte.
Yes, thank you. So I'm going to do the same as Pedro. I think I'm going to touch on several points, and then if we have time and if there's desire, we can dig a little deeper in any of those issues. So I think the research on biodiversity using genomics actually did something at the policy level, which is where I'm working, which is it made genetic and genomic data inevitable. There's so much of it now that it cannot be ignored. It has to be used for policy making, for global monitoring, etc., which for a long time, I think policymakers thought it would be useful, but they didn't know how, and so they sort of avoided the question. Obviously, measures of biodiversity are expanding. We're now talking about eDNA and microorganism sequencing. We're talking about a whole new branch of taxonomy that is emerging with the availability of this data, and that is hopefully going to be used, sooner rather than later for measuring biodiversity and taking decisions on the field, and what should be conserved, what areas should be conserved, how diverse they are, etc. There's another measure that I think has been talked for a long time, which is the intra-species diversity, which has been very elusive. You know, a few population genetic studies here and there. I mean, that's what I used to do when I was a researcher, but it's very difficult, it's time-consuming, it's expensive, and so you tend to select some very flagship species to do this. With the genomic era, these things are going to be more and more common, and so are actually going to become a tool for decision-making. So there's a whole sort of new world that's opening, I think, with the availability of this data. And in terms of policy making, governments in December took a very big commitment with the adoption of the Global Biodiversity Framework, the COP15. And so I think all this data is going to be in need, you know, very quickly. So this whole discussion on trickling down or trickling up, you know, all the data in some form that is digestible by other people than researchers is going to be more and more important. The other thing for researchers, I think, in terms of stakeholders, is opening the world of stakeholders a little broader for researchers. And so if we think about conservation, who are the most efficient people to conserve nature, biodiversity today are indigenous people. And so being able to sort of bypass government or bypass policymakers to start collaborations, technology transfer, capacity building, etc., between research organizations and local communities and indigenous people is actually something that is a really low-hanging fruit and an opportunity, I think, in your field of research. So some random thoughts, and we can dig into some later.
Perfect. Thanks, Charlotte. Far from random. That's very helpful. And now on to Tiina.
Thank you. I totally agree with the previous speakers that there is a urgent need for certain steps. For example, lack of common standards is one of those. So we really need to know how to take the sample and how to process it and how to use different kind of bioinformatic tools and so on, that we can have a reliable data. And when we were making our national eDNA roadmap, we made a survey with local stakeholders, and at least in Finland, there is a huge knowledge gap that those stakeholders, they don't know that what is doable today and what can be doable, let's say, within 5 or 10 years. So we really need this kind of information change, that what you can do today with these molecular-based methods and what is not yet doable. So this kind of information is really needed. And of course, because we are coming from a northern part of Europe, here in Finland and other northern countries, there are quite many lacks in those libraries, reference libraries with many organism groups such as diatoms. So these are gaps that we still need to fill before we can use these methods like in a full scale.
Thanks, Tiina. And now to Michael.
Yes. So, as the last one in this round, I agree with everything what you have been saying so far. And I would like to, so my two main things are trust and understanding. And trust in the methodology is linked to what Tiina just mentioned, the standardization. Actually, in Finland, Christian Meissner is based. He's working at the European level. I see her nodding. I've been working with Christian closely together to get the first European standard now out. It was beginning of this year for how to take water samples for eDNA analysis. Okay, so that's the first European standard we have. And I think we need much more of this standardization, because the stakeholders and the customers, the people who want to use the biodiversity information, they need to have trust in the methodology, and, you know, not like a random finding. You might send a sample to one laboratory and then to another one, and you get like different results. So that is not helpful. Unfortunately, for scientists, it's not the most thrilling thing to do standardization. But I quote Christian Meissner here. He says, don't publish, don't write papers, write standards. So there is kind of a gap between the science and the people who want to apply that. And as I'm also involved in this company, and we want to apply that, we see the big need, you know, for our customers that we can refer to standardization, which has just started, you know, because how to explain to a customer why they should do the analysis with us. Okay, what makes us different except from the price, perhaps? Yeah, so the quality is the one thing, and the trust in the method. And the other thing, the understanding has already also been mentioned, that the people, the stakeholders who are involved in getting those samples, in getting the results, in interpreting those results, they really need to know what the method can deliver and what it can't deliver. And yeah, that's very important. And there's a lot of education. Also from the company side, we have to do that, which is a bit difficult because we don't live from, or we don't make money with educating people. But I would say that that's a really important thing. And my final thing is, what in general I think is neglected also in the science is how to make these genomic methods combinable with standard methodology. Because the truth is that just using the genomic methods, they won't solve everything. You know, we need a combination, and this combination is rarely addressed in scientific work and in other developments.
Perfect. Thanks very much. And clearly there's a lot of commonality in the responses there. And I was just kind of reflecting on some of the key issues that came out there. So the critical importance of reference libraries underpinning all of this work, and that those reference libraries are high quality and curated. And then we kind of heard in many different ways there this kind of dual challenge of from a provider perspective, understanding stakeholder needs, but also at the same time helping stakeholders understand the potential of these new methods. And I think that point that you just finished with there, Michael, about this integration, not trying to solve all of the world's problems with one methods, but how do we integrate these data, these new data, with other information sources to build up a kind of holistic perspective of biodiversity. What ran through several of the responses there were the importance of standards, and this journey from proof of concept to operationalizing the method to standardizing and getting those standards in place. I like that. What was that phrase? Don't write don't write papers, write standards. I will remember that. That's a good point. But this is all part of this essential journey for building trust in a methodology that's repeatable and reliable. And I was just kind of reflecting on Charlotte's point about that. This is no longer a problem that can in any way be ignored. I mean, it's not that anybody wants to ignore it, but it's just has been a very difficult challenge. How do we handle this vast flood of new types of data? But we're at the point now of that journey of making this work, integrating these data into policy from both a species level and intra-specific level. And what's going to be key there is again, I'm just paraphrasing, that getting this linked through in a digestible fashion and getting data to people on the ground so that it works right the way through to the level of local communities. So there was a lot of commonality and related strands brought out in that initial set of thoughts. I'm now going to move to questions from the floor. And the first one is from Hanani Olivieri, and the question is, do you think defining priorities for species sequencing to fill knowledge gaps in molecular databases in order to move forward with eDNA biomonitoring is still an important issue. So this is about the kind of prioritization process of populating the databases. Anybody like to kick off on that one, Magda?
In fact, I reply with two not contradictory, but maybe complementary replies. I feel the need to have priority species, the species that are indicators for ecosystem health or deterioration. We have this kind of indicators in ecology, and for me, these are priority species. But at the same time, this is the other part of my reply. With environmental genomics, we are moving somehow from individual genomes to a more complex about the community. And for me, it's like very important now at this stage also to define standards for community, including all the organism, like from macroorganism to microorganism. So if we can also have, for example, what makes a forest a resilient forest? What is the diversity should be, or what level of diversity should have in its soil, for example, or how we can assess it. So moving from individual targets, priorities, to ecosystem or habitat, where we can define status with some diversity for the community. I don't know if I made myself clear, but for me now, for example, when we are working with forest restoration, we are working on this kind of standards. I don't know if I can call them standards now, but like studying this ecosystem diversity, this community together in a deteriorated forest, in a good forest, and in the perfect forest we would like to have, the ones that we call healthy and resilient, and compare this and see what are the groups that are coming up again, and that the group, which diversity and richness of diversity can be used as indicator.
Yeah, thanks, Magda. And I guess what I sort of got out of that there is this kind of almost contradictory element of the prioritizing the new data to help us understand the old indicators or the things we know about, versus the challenge of the floods of data being obtained now, telling us totally different things and new sets of things, and not purely prioritizing our activities around the things that we already know something about. Michael, from a the perspective of service provision for biomonitoring, the kind of reference library, what's in a reference library and what's not in a reference library must crop up a lot in that line of work. Do you have any kind of thoughts about this prioritization?
Yes, I think this making priorities, defining species is very important from the service provider's point of view, because then we know, okay, what the customer needs. It's a bit, often we explain people, okay, what you can do with the methodology. You can get a lot of taxa detected and so on and so forth, but it's a better understanding what's actually needed at the end of the day for conservation and biodiversity monitoring is really helpful for service providers. So a big yes on this question.
And Tiina, I'm just thinking with your eDNA roadmap for Finland, is this challenge will have come up again around how to most effectively operationalize eDNA biomonitoring at a national scale. And do you have any thoughts on this prioritization process?
Well, I guess in national level, first step is that it's really important to know that what we are now doing. For example, in Finland, many different kind of institutions are running their own pilots. So I guess it's really really important to know that what we are currently doing, and of course looked at what are our needs, what our stakeholder needs, and of course there are many things, for example, water framework directive based monitoring that I'm pretty sure that it will develop this way, that we will use molecular based tools quite soon. So I see that there are many levels where we need to this kind of knowledge, showing that what researchers are currently doing and what stakeholders needs and what are national level institutions doing this way, and how we can upscale it into international level, that we are like telling the same story. So there are many different questions on the table now, at least in Finland.
Perfect, thank you. And just a kind of final follow up on this one question, Elena, and just now moving to thinking about prioritizing which reference genomes to sequence, could you just get your thoughts on the process that the ERGA community has been through there?
Yeah, of course. ERGA focused first of all on the hotspots of Europe, but also it's focused on the species which are our conservation priority and bioeconomy, so economically important species. Because we know that, and we also heard today from really nice Swiss example, that resequencing data are really important for estimate effective population size changes over the time. And this is one of the important genetic indicator, which is also now included in the biodiversity framework program. So we really know what is happening with the population over the time and how we can also react in the conservation aspect. And through this, we also use the case studies which are connected with the conservation and the bioeconomy, but just not to prioritize them from the research part of view, but also from the stakeholder part of view, because we know that sometimes we also need to involve local communities, local stakeholders, because they also have some kind of priority in this aspect, and they would like to take care of their species of interest. So not just following the policy, maybe always, but sometimes really we need to think also about the local communities and citizens and society, because we know the genomic data also increase a lot of societal questions and ethical questions.
Perfect, thank you. So I'm just now going to move on to the next question. This is from Mark Blackster, and he says that for many of the stakeholder applications mentioned, it was not clear if absolute identification to species level is necessary or required, or even informative, and whether this getting species level identification or not makes a difference to regulatory authorities or stakeholders. Anybody like to respond on that one? Pedro?
Yeah, thanks for the opportunity. Well, for some of them, some applications, you need to have species level identification. If you are monitoring for the European Habitats Directive or Birds Directive, or try to detect a rare species, of course you need to know if it is that species or just a sequence that comes from whatever it takes. So knowing what's the species, at the very least in the case of species detection for conservation, it's absolutely necessary. And that I will make a parenthesis here regarding the standardization, because it's not only the standardization, is the detectability. Because sometimes we can do a standard and do precisely and repeatedly something that is not good. And so besides the standards, we need, or at least we need to incorporate at the standards, at least for species level detection, issues of replication, detectability, minimum levels of detection, because we know of many studies, for instance, in Africa, where they are selling detectability of red colobus or pygmy hippopotamus with technologies and sampling efforts that are totally insufficient for detecting those species with any level of reliability, though they are using precisely standardized methods. And so that's a very, very important issue. Going back to the question, the second part is regulatory agencies. Regulatory agencies, at least for the moment, require species level identification where the name of the species is associated with the sequence. That happens, for instance, with the Water Framework Directive, in which all the indicators and the groups need to be identified at species level. Of course, we can have estimates of ecological quality without the names. If we train a data set with previously known ecological indexes, we can train a system and to link directly the sequences to the ecological quality. But then it's like the artificial intelligence and deep learning. We know they have answers, but we don't know why. Why is that site better than another? So the ecological information behind the species name, it's not only a species name, it's a life history, it's an habitat preference, it's something that is predated by someone else and predates something else. So for most applications, having the names of the species increases the information. Of course, this may not be possible everywhere and always. If we go to the soil, or to microbiomes, or to very regions that are less studied, not going to the species level may be enough, but I would say it would always be good to have a species name behind the sequence. And I think this is all for the moment.
Thanks, Pedro. Mikhail?
I completely agree with Pedro. Species detection can be very important, but just one comment on the standardization, because there seems to be a misconception a bit, because standardization doesn't mean it's exact protocol recipe you have to follow and you can't change it. The current standard we have created for how to take environmental DNA from water samples just gives you the minimum requirements and the general outline how to do this work, okay? I completely agree that you need to have a suitable technology starting from how you take the samples, how you preserve them, how you treat them in the laboratory, up to the data interpretation. All this is kind of in need for standards, but standards are not recipes which are written in stone, but they give a framework so that the people following these standards can assure a certain level of quality.
Charlotte, were you wanting to come in there?
Yeah, thank you. Yeah, I think I'll actually answer linking also to the previous question maybe a little bit. So I agree with everything that people have said, obviously, and I think this species, absolute species identification, I think that was the term, is a bit of a term that I think even within the scientific community can be defined in different manners. So I think a standard actually would be good on that. But I also want to talk about, first of all, when we start talking about microorganisms, I think you can't name them. They get to the species level. There is no naming of them as of today. There's so much data and so much flooding and so much to understand still about those organisms. But they are very important for the measures of biodiversity. So I think the standardization of the maybe not naming but quantifying them actually is very important for policymakers. But in terms of the species, I actually think it's very important, first of all, for policy. I also work in the same unit that regulates implements the Nagoya Protocol at the CBD, and so identification to species level is very important for policy makers. And I think all the work that is being done, in Europe, and I see in this panel that talks about data quality and standardization, what does it mean, etc., is really important. And I think in terms of capacity building and technology transfer, the example of sampling in Africa being of sub-quality compared to the samplings that you all are able to do in Europe, actually goes to the problem of baselines and being able to prioritize what species and what areas have to be sequenced, because you can only protect and work on what you know. And so the very non-sexy work of scientists to actually go out there and sample and register the species and just sequence environments and have high quality sequence for what is there globally is, I would say, just as important for the conservation of biodiversity as improving, your own methods and standardizing things within Europe. So.
Can I just add two very basic examples like IUCN and CITES. Like for IUCN, if you want to see what is the status of the species, we need to really have one species and identified to see its distribution, its population, etc. So we cannot, as Charlotte just said, we cannot protect what we don't know. So just for a basic thing for IUCN, it's a species level identification, but also for forensic application for CITES, for example, if you want to control what's happening. Sometimes you have one species that is very, very widespread and another one in the same genus, very close to it, is completely rare. So if we don't have this individual species identification, we can just mess up the conservation of this species, and CITES cannot work with this.
Perfect. Thanks, Magda. And just as we're kind of wrapping up that particular theme there, there's a related question that's come in from Katie Clark from Natural England, and she's basically saying it's come back to this point about standards. Should we be looking for setting set methods for monitoring groups of taxa, so taxon specific, really tightly defined protocols, or setting the kind of key principles that all should use? And Michael, I guess that's just kind of following up on your earlier comments.
Yes, it's about the key principles, and it's not easy as the methods are rapidly developing. And so the plan, I think the international plan is to do it stepwise. So we started kind of with how to take a sample in the water now. The next step will be a single species identification, and other steps will be in meta-barcoding, how to create standards or tests where laboratories could test or could be tested how well they are doing the work. So it's kind of, there's questions involved: who will organize these ring tests, this accreditation, who will compile these DNA samples so that the laboratories can be tested, and so on and so forth. So it's quite a complex thing. And in general, I would say it would be good if not everything which comes out in science and is published as a paper as the newest and latest and most sexy approach should be followed at this stage. So for the practical application, it would be useful if we step back a bit and try to build solid protocols, processes, so that the stakeholders, the people who need the biodiversity information in the end, can work with. Okay. As a company, you can't cope with all the new developments, and this biodiversity genomic field is so rapidly developing. Even as a scientist, you have problems. There's so many papers coming out each week, like following the field and be up to date, and just think about how it feels from a service provider if you want to try to implement that.
The downsides of repeated and rapid innovation. So, okay, so changing, I was just going to change tack now. The question from Rob Waterhouse, and he says, we've heard several times today the idea of decentralized or distributed efforts in biodiversity genomics. And what do the panel think are the main advantages of this approach versus the biodiversity equivalent of CERN, the kind of mega infrastructures? Elena, are you happy to to kick off on that?
Yeah, I would like also to add what was Michael said regarding the approaches, which and we know that many questions in conservation can be answered also with some classical approaches, which are not maybe in the front of the genomic research now. But keeping in mind also the diversity of Europe, and we know that we have, if we follow European Commission division into the strengthening and widening country, we still have this kind of division and different financial aspect. And we know that maybe in strengthening country, we have really already the infrastructure research and infrastructure, the financial support, which is not so much developed in the widening country. And I think what was Charlotte was saying, the capacity building, it's coming really, really important throughout the Europe. So maybe this standardization, the standardization aspect we were discussing today, we should think also this kind of diversity across the Europe into the account. So having the approach when we can really answer this kind of conservation question or any other question which we are interested now in prioritizing, but also keeping in mind that diversity across the Europe, can we make this in the most cost-effective way? Should we really use for everything the most modern, the new approach, or we can do something in the most effective way that also stakeholders all across the Europe can have this approach and get the answer they really need to for the biodiversity monitoring. So I don't know if I answered to the question, but this is something we as ERGA, as a European Reference Genome Atlas, always have in mind, the diversity across the countries.
And maybe, Charlotte, if I could just kind of ask from a CBD perspective, this kind of the views of this kind of distributed systems versus kind of large central points.
Yeah, absolutely. So I think, you know, as an organization that works at the global level, we're always interested in centralized data, because then we don't have to standardize and aggregate it ourselves. But there's a lot of value in specialized data sets and specialized funding, because the decentralized sort of approach also allows researchers to focus on areas that maybe if they had to compete, you know, if you have to compete ferns of Africa with elephants, chances are elephants are going to win the jackpot in terms of the funding and the attention and etc. But if you have specialized approaches and more local funding, then you're able to really dig into things that are not getting global attention. But I think this is where the question that, you know, everybody, Michael and others have been talking about, which is then you have to be able to aggregate that data. Maybe not all of it, but at least some of it. And so I think that has to be done prior, deciding which ones are which data has to be standardized across, you know, specialized data set or repositories or research organizations, so that we can aggregate them later and we don't have to go back and modify that data and realize we can or we cannot, and it's so much work. But if we do that off the bat, then we add value to the decentralized initiatives as well. That's the way we see it.
And there's of course a very practical benefit to these decentralized efforts in biodiversity genomics when it's in terms of the data generation, of the minimizing the kind of challenges that go with the movement of samples across international boundaries, and essentially and also around this the democratization of access to technologies. Okay, so just flicking through the list of questions here, and there's one from Veronica Mrazek asking, do we have any thoughts how in practice we can most effectively communicate these complex data sets to local communities and stakeholders? Is is this best handled by individual researchers, or is there a need for specialist science communicator roles within the research teams? Who wants to take this? So, Charlotte, then Pedro.
Sure. So I'll jump in here. Thank you for your question. So I think science communicators are really rare breed. And so this is definitely best handled by researchers themselves. And in terms of stakeholder involvement, there's several aspects that have to be taken into consideration, I think, and the research community can learn. It all depends what your research is for, right? It depends what the goal of your research is. But if the goal of your research is to help a certain area, for example, or a certain local community, or taking advantage of traditional knowledge that might exist, then those communities have to be involved before the research even starts, right? So they know what they need. They know better than you what they need. And so they have to be involved in the design and in the question and in the goal of the research project. And if you involve them from the beginning, then sure, you can build capacity around them being able to do genomics one day, but also you can involve them in understanding why you're using that tool, right? Because genomics, genetics is just a tool to answer a question. And so the important thing to them might just be the answer of the question, to be able to help them in what they need to conserve biodiversity. I'm going to put, I don't know if I can, I'm going to put a link actually to community protocols that exist. There are a lot of international regulation, particularly if you're going to work with indigenous people or on indigenous territory, that you have to be aware of. But there's also a lot of local communities that might not be indigenous that have created community protocols. And so researchers should always inform themselves of what's already happening, what are already the standards for being able to come in a community and using their biodiversity and doing research around it. So I'll just post a link on it.
Thanks, Charlotte. And Pedro?
Well, I will take this to digress again on the need for species names, because it's always difficult to communicate conservation and conservation needs to local communities. And we are working in Europe, but also a lot in Africa. And you have to show what is the importance of some areas, and some areas are more important than others, and all that kind of thing. And if you go in a like we are doing large-scale surveys in places like Angola, Guinea-Bissau, Namibia, now Equatorial Guinea, and we do eDNA sampling, we get the sequences, and then we get them a name, and then we say in this forest there is A, B, C, and X, and in other forests there is Z, D, and V, and we can link with actual things that people know and they are familiar with. If I say there are more sequences in this forest than in another forest, and so this is the proper forest to be protected, that will cause a huge communication problem. So it's always difficult to communicate conservation. It's even more difficult with genomic tools, and then we have to translate to the local communities, and the translation and the mediator are the species names, are something people are familiar with.
Elena?
Yeah, I would like to add something here. Also, across the Europe, we really have a rising of the citizen science project. And I agree with Pedro that it's very difficult to communicate with the stakeholders, with the citizens, with the society. But through the citizen science and through these kinds of citizen science initiative, we can really use the 10 citizen science principles, which are really important, and really define that we are not using a community just as a sample collectors, but we are collaborating with them. We are sharing the data. We would like to hear their need, their interests, So these are something we are following now, these ethical and societal needs. Of course, we need the scientific communicators. We even need some contributor from social science so that they can give us as a researcher working in genomics and natural sciences. Maybe we do not have these kind of skills, but I think that we should use this opportunity to involve citizen science, even in genomics, more and more active. Of course, together with the other data, which are important ecological data, which can be also put together with genomics by using citizen science.
Thanks, Elena. Tiina?
Thank you. We have a quite good example on Finland. We have this kind of eDNA network, which is open to everyone. It has been running almost 3 years now, and we have, same online meeting 4 times per year. We have stakeholders, researchers, citizens who are interested in these methods, and it's really quite easy way to share the newest knowledge and what we have been doing and the newest results, and at least we have been feeling this kind of joy and enthusiasm that those stakeholders, they are really willing to understand more and hear more about these methods. So these kind of online meetings are actually quite easy way to really inform those stakeholders what is going on, and of course then they can provide what kind of needs they have and so on. So just a good example. It doesn't need to be too hard to organize.
Perfect. Magda, then Mikhail?
Yeah, I just want to continue at what Tiina just started by saying, but if I speak about, for example, Africa or Latin America or the Middle East, the best way to communicate the message with those community is also can go through education and training, and we can see the opportunity of advanced sequencing technologies that are becoming more and more affordable and democratized somehow to work on this. And when people see that their own researchers are able to do this, it will not be perceived as a rich world biodiversity problem. They will know about their own biodiversity, they will care about it, and they will be keen to go and deep further to research and give names to the species they are discovering. So also using the local languages, the local resources, the local communicators is a key to get the message to them.
Mikhail?
Yeah, from the point of view of a service provider, I would say that definitely you would need people who are specialized in communicating those results. Because those people who are doing the lab work, for example, are not trained for that, and they are sometimes not the best ones, to bring this really to the end user. Okay, so from in our company, for example, we have a dedicated team who is just dealing with the customers, explaining the results to them. So we have no people from the lab directly talking to customers, because this can also cause confusion. And it builds, or this goes a bit into the direction of building understanding and trust. And it can be very helpful to have people who are specifically trained, set up for explaining the stuff in simple, understandable way.
Thanks all, and I see we're just about to run out of time. So if I could just, we could just quickly go around with a chance for any last closing remarks from the panel members. Just literally a sentence on your kind of key take-home messages and reflections from the discussion. Magda?
I will reiterate again the importance of local knowledge, of educating and training people on the ground everywhere to make this science more democratized.
Elena?
I think that we should build a trust between the stakeholders and researchers. The genomic is something which benefits society.
Pedro?
I think the time was too short for discussing all these complex issues. And I'll be looking forward for other opportunities to meet and discuss these kind of things, which are really, really important.
Charlotte?
Thank you. I echo Pedro on future opportunities, hopefully. My final thought, I would say, is broadening the stakeholders involved in the research will only maximize the impact of that research and those results.
Tiina?
I think we are already on a right way, but I cannot enough underline the importance of standardized methods. And good news according to that I think that this eDNA method standard is now maybe now it's approved in SEN standardization, and hopefully quite soon Christian Meissner will start with ISO standardization. So it's really important that we have this kind of common understanding that and minimum level standards which to follow. Thank you.
And Michael?
It's a great time and a great tool in hand of what we have in hand for assessing biodiversity. We need it. It's great that you have invited also someone who is doing service provision. Don't forget the service providers. Thank you.
Perfect. Well, we've run out of time. I take the point that we need much more time to discuss these complex and wide-ranging issues. But I just want to finish with a big thank you to the panel. We covered a lot of ground there, and that was some fascinating discussion. Thanks to folk feeding the questions in, and all of the speakers in the day for this Biodiversity Genomics Europe Day. Thank you very much.